genetics. And the subsection of genetics is inheritance. And further information, you can go as always to these separate articles: genetics, Mendelian inheritance, and classical genetics. All right, so genetics is the scientific study of inheritance. Yes, Mendelian inheritance specifically is the process by which genes and traits are passed on from parents to offspring. It was formulated by Gregor Mendel based on his work with pea plants in the mid-19th century. Mendel established several principles of inheritance. The first is that genetic characteristics, which are now called alleles or gene variants, as I described earlier, are discrete and have alternate forms, e.g. Purple versus white or tall versus dwarf. And each is inherited from one of two parents, based on his law of dominance and uniformity, which states that some alleles are dominant while others are recessive. An organism with at least one dominant allele will display the phenotype, which is observable traits of that dominant allele. Exceptions to this rule include penetrance and expressivity, or penetrance and expressivity. And now, let me just note, as I already went over allele, we'll just go over it again because this is in line where, with the topic that aligns with it. So an allele, modern formation from Greek allos, other, is one of two or more forms of a gene variant, or of a given gene variant. Yeah, and this definition, interestingly, I copied and pasted this actually, I believe last year or earlier this year. I believe it was possibly last year. If I go back to this one here, if I go figure allele and search up this one all the way back to the earlier time I just referenced it, this one I copied and pasted. This one's actually I believe yesterday or 2 days ago. So the term allele, modern formation from Greek, this one allos or other, denotes the variant of a given gene. So Wikipedia just changed slightly the wording. And also note this part. So in genetics, it is normal for genes to show deviations or diversity, and all alleles together make up the set of genetic information that defines a gene. Yes, you have variants to that gene. So anyways, so just gene variant. Alright, so now let's go over this dominance set right here. So based on Mendel's law of dominance and uniformity, which states that some alleles are dominant while others are recessive. So dominance is a phenomenon of one variant allele of a gene on a chromosome masking or overriding the effect of a different variant of the same gene. So for example, one is giving blue eyes, other one's giving red eyes or brown eyes. The brown eyes is going to win because that has the dominant allele at that same spot where the collection of them is the gene for eye color, for example. Yeah, so let's read that again. So dominance is the phenomenon of one variant allele of a gene on a chromosome masking or overriding the effect of a different variant of the same gene on the other copy of the chromosome. The first variant is termed dominant and the second recessive. And there are several types right here. We could make this, I'll make it like this and I'll just zoom in when we need to. So here is an autosomal dominant and autosomal recessive inheritance, the two most common Mendelian inheritance patterns. An autosome is any chromosome other than a sex chromosome or chromosome. We'll get to those in a bit. So autosomal dominant right here. So if you have this one right here. Yeah, this color right here, this is just white. Let's look at the blue one here, this dark blue. That is the affected one. So for example, let's say you had this dominant one right here is this one here. So then she would have the blue eyes or whatever. So then because she has one of the dominant gene, this one has a recessive one. So he's not affected by that dominant one right there. And I just put a note here. So note that the carrier parents carry the recessive genes but are not affected unless both gene copies are recessive. Yes, so in this blue one, this is a autosomal dominant. So for example, this one's not affected, doesn't have those two genes. It doesn't have any of this gene. And this one right here is affected, has a dominant one, so it has, let's say, blue eyes. Or blue skin. Alright, and then they have kids, and then what you're going to have is a chance of getting, of this getting inside, or you're going to be sharing chromosomes, so you could have this one, and this one in a child that's over here, and you're gonna have And then what you'll have is this one. Yeah, that one and this one, for example. They can line up and you get this one. Or you could have, instead of that, you could have had this one and this one and this one. And they line up and get that one there. Or the last one, you can have something like this one and this one. And you get this one here. This is unaffected. Yeah, so only two of these would be the... Yeah, two of these would have the skin color. And the other one is not carrying that specific skin color gene, the blue one. And then this is autosomal recessive. So this is a recessive one. So this one here is recessive. So what's going to have is, and then there's also no carriers here that aren't affected. So this one is not carrying any of it, and it's not affected. This one is affected and carrying, so it's just the same thing. And this one is, and this one isn't. And then now if this is autosomal recessive, this one is not going to be affected. So it doesn't have skin color, but it's light blue to indicate that they both are carrying a recessive gene for the blue skin color. Yeah, so in this case, what you're going to have is you're going to intermix. This one would go with this, so you have this is unaffected. And then what you'll have here is the next one is the next one could be this plus this one. That's a carrier because it only just has one, but it's not going to be you're not going to have blue skin color, but just make it light blue. And then this one over here, you could have this one and this one you get over here, and still nothing. Oh, just a carrier, and it's the same as their carrier parents. And then the last one, if this recessive has a recessive gene and they link up, you're going to get blue skin color most of the time, or high percentage of the time. So fascinating stuff. So let's continue further. And now let's look at the exceptions to the rule, and that is penetrance and expressivity. So penetrance in genetics is the proportion of individuals carrying a particular variant or allele of a gene, the genotype. And we'll get to genotype soon. That's over here. And right. Yeah, so it's the proportion of individuals carrying a particular variant or allele of a gene, the genotype, that also express an associated trait, the phenotype, so the actual visible characteristics, so like blue skin, for example. In medical genetics, the penetrance of a disease-causing mutation is the proportion of individuals with a mutation who exhibit clinical symptoms among all individuals with such mutation. For example, if a mutation in the gene responsible for a particular autosomal dominant disorder has 95 penetrance, then 95% of those with the mutation will develop the disease, while 5% will not. And I mean, inside this raises the question of what actual causation is. What the actual causation is, because what's happening to these 5% that's not? And what is the process of getting, well, you have the person with the supposed mutation, and then what's the process, or how do you, and especially the psychological process that they have to deal with if you tell them, okay, you have a 95% chance of getting this disorder. Or if it's something at a young age that they have, then the question is, well, why is it that these 5% don't have it? So there's something else that could be at play. So yes, just food for thought. And the genotype of an organism is its complete set of genetic material. However, the term is often used to refer to a single gene or set of genes, such as the genotype for eye color. Then the other exception is expressivity. Expressivity, is the degree to which a phenotype is expressed by individuals having a particular genotype. So it's the degree to which a phenotype is expressed. So in other words, expressivity is related to the intensity of a given phenotype. It differs from penetrance, which refers to the proportion of individuals with a particular genotype that actually express the phenotype. So in other words, penetrance is a expressivity of 0. And then if you have extremely dark blue eyes, it can be that could be high, or 100%. If you have light blue, it could be 50%, for example. So expressivity is the intensity of the phenotype, or the observed characteristics. All right, so let's continue further. So Mendel noted that during gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for each gene. And this is stated by the law of segregation. Very interesting. So during the formation, instead of having two alleles for one gene, each one will have just one. So alleles for each gene segregate from each other so that each gene has just one. So yeah, you're just going to have one on each parent will have one. No, I mean the gamete will each have one. And then when you intermix with the other gamete, you're going to form now two alleles on the full chromosome pair, such as over here. Yeah, such as over here. So BB, so you have two of these together there, but then each one of these has one. That's very interesting. So MES note, this is the next part here. So heterozygotic individuals produce gametes with an equal frequency of two alleles. And so MES note, zygosity is the degree to which both copies of a chromosome or gene have the same genetic sequence. In other words, it is the degree of similarity of the alleles in an organism. So, for example, right here, so if you have this pair of chromosomes right here, and these both copies. So here's the capital B and B. These are homozygous, or homozygous. That's capital BB. So they're exactly the same. So there's B and B right there. And then this other one is the other gene, lowercase b and lowercase b. That's also homozygous, and that's also connected. But then if they're different, so now if this is different, this is going to be capital B and B, and this one's over at this location is lowercase b and capital case B. This is heterozygous. Or heterozygous. This individuals with an equal, or heterozygotic individuals produce gametes with an equal frequency of two alleles. So there's two alleles, equal frequency. But this one here is just a homozygous, just to illustrate. Yeah. This illustrates zygosity. Very interesting. So finally, Mendel formulated the law of independent assortment, which states that genes of different traits can segregate independently during the formation of gametes, i.e. genes are unlinked. An exception to this rule would include traits that are sex-linked. Test crosses can be performed to experimentally determine the underlying genotype of an organism with a dominant phenotype. Yeah, so these ones were above here. So genes that of different traits, such as eye color and hair color, or etc. These segregate to the daughter or child, to the child independently of the gender. But then this one over here, this is the sex-linked ones are an exception, which depend on the sex or gender. So let's go over here. So MES notes, sex-linked describes the sex-specific patterns of inheritance and presentation when a gene mutation or allele is present on a sex chromosome or allosome, rather than a non-sex chromosome, which is autosome. Yeah, in humans, these are termed X-linked recessive, X-linked dominant, and Y-linked. And the inheritance and presentation of all three differ depending on the sex of both the parent and the child. This makes them characteristically different from autosomal dominance and recessiveness, which don't depend on the gender or the sex. It's very interesting. Let's continue further. So a sex chromosome, also referred to as an allosome or heterotypical chromosome, guanosome, gonosome, or a heterochromosome or idiochromosome, is a chromosome that differs from an ordinary autosome in form, size, and behavior. The human sex chromosomes, a typical pair of mammal allosomes, determine the sex of an individual created in sexual reproduction. Autosomes differ from allosomes because autosomes appear in pairs whose members have the same form but differ from other pairs in a diploid cell. Yeah, whereas members of an allosome pair or sex chromosome may differ from one another and thereby determine sex. Yeah, so the autosomes, the non-sex chromosomes ones, they differ from allosomes because autosomes appear in pairs whose members have the same form, but just differ from other pairs. But allosomes differ from each other, and then in that way they can determine sex. All right, let's continue further. So an autosome is any chromosome that is not a sex chromosome. The members of an autosome pair in a diploid cell, the members of an autosome pair in a diploid cell have the same morphology, unlike those in allosomes, which may have different structures. The DNA in autosomes is collectively known as in autosomes is collectively known as AT-DNA or AU-DNA. For auto or the AU, the AUTO. For example, humans have a diploid genome that usually contains 22 pairs of autosomes in one allosome pair, which is 46 chromosomes total. Yeah, so you have 22 pairs of autosomes times by 2, that's 44, and then you have a pair of allosomes, so that's plus 2, so 46, or 23 times 2. The autosomes are labeled with numbers 1 to 22 in humans, roughly in order of their sizes in base pairs, while allosomes are labeled with their letters. By contrast, the allosome pair of 2 X chromosomes in females or 1 X and 1 Y chromosome in males. Yeah, so in other words, the allosome pair may differ from one another, then thereby determine sex. So that's the key point right here. And notice right here, so if you have an X and an X, so if you have 2 X chromosomes, that's in females, or if you have 1 X and 1 Y, that is male. So if you have a Y, that's a male. Unusual combinations of XYY, XXY, XXX, and then you have XXX, and then this other one, XXXX, or XXYY, among other combinations, are known to occur and usually cause developmental abnormalities. The X chromosome is one of the 2 sex-determining chromosomes or allosomes in many organisms, including mammals. The other is the Y chromosome, and is found in both males and females. The X chromosome in humans spans more than 153 million base pairs, which is quite fascinating. The Y chromosome is normally the sex-determining chromosome in many species, since it is the presence or absence of the Y that determines the male or female sex of offspring produced in sexual reproduction. In mammals, the Y chromosome contains the gene SRI, or sex-determining region Y protein, which triggers male development. The DNA in the human Y chromosome is composed of about 59 million base pairs, and the Y chromosome is passed only from father to son. So it's a sex-linked trait or inheritance, and this is opposite of the autosome. And as stated here, so an exception to this rule of the law of independent assortment would include traits that are sex-linked. So it depends on the gender of the child and the parent. Whereas in most other cases, they're unlinked. So it doesn't matter if it's a mother or it doesn't matter what the sex or gender of the parents and children for pretty much most of the other traits. And usually such as eye color and so on. But then specific traits that are sex-linked, such as the actual gender of the individual. And let's go further. Let's read up on this XY sex determination system. So the XY sex determination system is a sex determination system used to classify many mammals, including humans, some insects such as this Drosophila or Drosophila, and then some snakes, some fish such as guppies, and some plants, the ginkgo tree. These ones all use the sex determination system, the XY one. In this system, the sex of an individual is determined by a pair of sex chromosomes. Females have 2 of the same kind of sex chromosome, XX, and are called the homogametic sex. Males have 2 different kinds of sex chromosomes, so the XY, and are called heterogametic. And these are also pronounced, I believe the correct pronunciation or the more common one is homogamatic and then heterogamatic. I've been saying gametic, but I just googled the pronunciation and it states homogamatic and heterogamatic. Interesting. Then I'll continue further. So here is a Drosophila, or a Drosophila, which is a fruit flies, and their sex chromosomes, like here. Make it smaller. So the female has both XX sex chromosome, and then the male has X and an X and a Y. So the Y is typically smaller. So if you compare this one here, is 59 million base pairs for humans, and then the X chromosome is 153 million base pairs. So almost, that's about almost 3 times bigger. And notice the different characteristics of this one. Of this female looks like this. It's a bit bigger than the male one, and this looks like that. Note the stripe pattern in this animation or this image. And continue further. Now this is right here. This figure shows the pollen the pollen cones of a male ginkgo biloba tree, and it's a dioecious species. Notice the cones there. We'll go over pollen and later in this video. Notice the pollen to indicate it's male, and then there's the ovules of a female ginkgo biloba. Or biloba. And dioeci, from the Greek, this word 2 households, adjective, and its adjective form, which is a modifying a noun or person, place, or thing, is dioecious. So like a dioecious species. So dioece is a characteristic of a species, meaning that it has distinct individual organisms that produce male or female gametes, produces the male, the pollen produces the male gametes, and the ovules produce the female gametes, either directly in animals or indirectly in seed plants. Yeah, and I believe this indirect or direct, for plants, indirect is it produces pollen, which contains that gametes inside, and so on. Close this. So these ones produce the pollen, which contains the gametes. I believe that's why they're using the term indirectly, and I just put that here. So male plants produce pollen, which contains gametes. So the pollen goes into the female plant, and then it eventually gets into the egg, and then releases the sperm cells or the gametes or the gamete, and then inside this egg one. And hence the word indirectly there, as opposed to animals, they produce it directly. So they produce, the male and female both produce the sperm and egg directly, and then they fuse. This one this is indirect one. The male plants produce pollen, which contains gametes. So then the pollen goes in, and then it releases. Anyways, going further, dioecious reproduction is biparental reproduction, so it's two parents. Dioece has cost since only about half the population directly produces offspring, and that is the female population. Yeah, compare this with bacteria or algae, which just automatically duplicate. Pretty much all of the cells can duplicate. When this one, dioece has cost, so they only have half the population producing offspring. And going further. Continuing further, humans are diploid organisms, normally carrying two complete sets of chromosomes. Normally, that's interesting. So normally carrying two complete sets of chromosomes in their somatic or body cells, two copies of paternal and maternal chromosomes, respectively, in each of the 23 homologous pairs of chromosomes that humans normally have. This results in two homologous pairs within each of the 23 homologous pairs, providing a full complement of 46 chromosomes. So 23, each one has a complement, and they're all duplicated. The somatic ones are pretty much almost identical, or just in the same form, but then the XY ones are the ones that differ greatly. Continue further. So here is a graphical representation of these chromosomes. So graphical representation is of the idealized human diploid karyotype, which is ordering of chromosome pairs, showing the organization of the genome into chromosomes. This drawing shows both the female XX and male XY versions of the 23rd chromosome pair. Chromosomes are shown aligned at their centromeres. The mitochondrial DNA is not shown. So centromeres, where they connect to each other. So here's the chromosome 1, and it's listed generally by size. It's in the from biggest to smallest by base pair generally. So number 1, 2, and there's 3. And notice how they're all very similar to it and almost identical. So 10, 11, all the way to 22. And now you have the 23rd one, which is XY, which is male, or XX, which is female. And the mitochondrial DNA is not shown. This is just the chromosomal DNA. Nuclear DNA. Okay, that's the nuclear DNA right there. And now here is this figure right here. It's more representative of how it actually looks in real life. Let's just see. This is from Socratic.org. And I believe this is a micrograph. So the figure shows karyogram ordering from a karyogram, ordering from a micrograph, and karyotype is just ordering of chromosome pairs, just the ordering. This is ordering from a micrograph, and this is a colored micrograph, so that's the shapes of the actual chromosomes. For a human male, where you can see set of 23 pairs of homologous chromosomes, say a human male. And notice how the real one, this one, the Y one, is much smaller than this X chromosome. And notice the shapes there and so on. It's pretty much, they're very similar, but they could be different shapes and so on. Continue further. And notice the size difference, how it gets big all the way to small. So big all the way to small, and then you have the XY, which are separate. And continue further. Here's another karyogram of human male using GIMSA staining. Yeah, or Giemsa staining, which is a nucleic acid stain, and yeah, just leave it at that. So there's another karyogram here, and notice the different shapes and so on. It gets from the biggest all the way to the small, and there's the XY chromosome right there, or XY chromosomes, yeah, or pair, etc. Continue further. So X-linked recessive inheritance, and just go over here. This one we discussed over here when I discussed the inheritance types. So the sex-linked inheritance describes sex-linked describes the sex-specific patterns of inheritance and presentation with a gene mutation or allele is when a gene mutation or allele is present on a sex chromosome, allosome, rather than a non-sex chromosome, autosome. So in humans, these are X-linked recessive, X-linked dominant, and Y-linked. And there's three, and this is because of the different forms of the X and Y. So they're not almost identical like the typical autosome. So these ones are pretty much identical, and these ones are not. So it's going to have three as opposed to the four that we're used to. Let's continue further. So X-linked recessive inheritance is a mode of inheritance in which a mutation in a gene on the X chromosome causes the phenotype to be always expressed in males. Yeah, and who are necessarily homozygous or homozygous or hemizygous. Yeah, for the gene mutation because they only have one X and one Y chromosome. Yeah, so if it's on the X one, so it's a male, it's always going to be there. But for females, because this is a recessive, it needs it on both of the chromosomes. And in females who are homozygous for the gene mutation, see zygosity, females with one copy of the mutated genes are carriers, because it has two X chromosomes. Or two X chromosomes, so it needs to have it on both because this is a recessive. And a dominant we would just need it on one. So a chromosome in a diploid organism is hemizygous, or homozygous when only one copy is present. For organisms in which the male is heterogametic, such as humans, almost all X-linked genes are hemizygous in males with normal chromosomes because they have only one X chromosome, and few of the same genes are on the Y chromosome. This is interesting. So just few of them are on the Y chromosome. So almost always it's going to be hemizygous or homozygous because because it only has one of the X chromosome, so it doesn't need to worry about the Y one. So here's an illustration of the X-linked recessive. All right, so X-linked recessive inheritance. Yeah, so X-linked recessive inheritance, and so we have two scenarios. Again, it depends on the gender of the parents, what they have, as well as the offspring, which gender or sex they are. So if you have an unaffected father who doesn't have any of this X-linked recessive gene, so we'll just have this X1 has just a blank one there, and the female is a carrier because she only has one, and because this is recessive. For a female, it needs to have both. So this one has one is the carrier, so she's carrying that gene, but then the other side of the chromosome isn't. So then what's going to happen is the offspring is going to be you're going to have this intermix with, and this X1 is going to go with, well, actually, no, the male one doesn't pass that X1. It passes the Y. So it's always going to pass the Y to every single one. No, every single male. So the Y is going to go to every single male. But the X1 is going to pass only to the female. So you're going to have this one goes over, let's say it goes over, just put it over here. And then this right here, let's say it gets it over with this. So that one goes there, and this goes, it swaps over, and you get the female. So that's the X and Y. So it can be a carrier daughter as well. And then the next one is, you're going to send this part of the X of the female over to here, and then this one's going to go there to get the female. So this one's going to be unaffected, doesn't have the gene, but the male one has just one, and that's going to be affected. So it's going to have that blue eyes or whatever it is. So this one is affected. Yeah. So then that one has it, and this one, because there's no other, doesn't have the same gene on the other side or allele of it on the Y. This one has another allele. It's not this gene, so it's recessive. So it's not going to appear, but she's going to carry that. So note, a few carriers may be mildly affected due to skewed X inactivation. And hence here, so it might have low intensity there. So a few carriers may be mildly affected because it's Yeah, so it's not always going to be fully identical. This fully gene, or this allele is the affected one, or carrying this one, and then it's not going to be completely offset by this one. It might be a skew distribution or activation, and so on. And now here's the other. Let's say here's another example: is if you have the affected father. So this one has unaffected father and carrier mother. This one's affected father and unaffected mother. So she's not carrying. This one is affected and hence carrying. And then what's going to happen is so this X one right here. So if it gets to the female, or let's just say it swaps off with this X right there, this would be a female over there. And then the next one is going to be, let's just erase this. Then the next one's going to be, let's say if you have a Y, and this one here, this X is over here, so nothing's there. And then the next one over here is going to be a Y, and the other X. And that's going to be a male that's unaffected. And then the last one is if you have this X right here, and then this other X. It goes over to here, and this is going to be a carrier daughter. So none of these are affected or affected largely, unless by the skewed inactivation. Yeah, so basically none of these, there's just going to be carrier, because none of the males have it. Because this the male can't pass the X to the male. That's only from the females, because they all swap over. So it's going to be an unaffected son, but the carrier daughters. So very interesting. So now let's look at X-linked dominant inheritance, sometimes referred to as X-linked dominance, is a mode of genetic inheritance by which a dominant gene is carried on the X chromosome. So basically the opposite of the recessive. And now it's pretty much, if it shows up anywhere in male or female, it's going to be affected. So if you have this example, unaffected father and affected mother, and then you're going to swap it over. So this one would be XY. This Y goes over here, and this X goes over there. And then this one here would have the X here, and this X. So this one is going to be affected. And the affected son would be this one here and this. And then the unaffected daughter would be this and this part. And this unaffected. Yeah, so very interesting. And note right here, as a note, some X-linked dominant disorders are embryonic, are embryonic lethal in males, so as they're getting formed, in the embryo, and most affect females less severely. Yeah, so X-linked dominant affect the male more than the dominant disorders are embryonic lethal in males when it's a disorder or abnormal alteration, etc. And then those will affect the males more than the females. And it's again because that Y usually doesn't have the same genes as the other side. Yeah, and then the result of this one is going to have two affected an affected daughter and affected son, and unaffected daughter and son. There, that's going to be for this scenario. But now if you have the father with the gene, so on the X, so affected father and then unaffected mother, these are again dominant genes. This one is X is XY. What you're going to end up with is this X goes over to here. This one is X over here. And then the Y is over this one, unless you'll have a Y over there. And then this X goes to, let's say it goes to here, and then it gets to the other X. And likewise, this Y goes over here, and it could be the other X over there. So yeah, you can just swap it over there, and what you're going to end up with is two affected daughters and no affected sons, because it doesn't have it. You can only pass the Y to the sons. So yes, that's again sex-linked. It's very interesting stuff. Let's continue further. All right, so now let's look at Y-linked inheritance, also known as holandric inheritance from ancient Greek. This holos, whole, and this andros, male. So it describes traits that are produced by genes located on the Y chromosome. It only occurs in males. Yeah, so you'll have here Y-linked, so affected father has the Y. So it needs to it's the only one that has it, so it's only on the Y. And then, yeah, so only over here the Y, and then the unaffected mother doesn't have the Y. So then basically this is going to go to the male, and this one's going to go to a male. Well, here's always going to be on the male, on each one of these, but the females, it's always going to be unaffected. You're always going to be swapping the X with either X that doesn't have it, and goes down there. Yeah, so you're just going to end up with two affected sons, identical or just very similar to this, and then you'll have unaffected daughters right there. So the genes just get passed on to the males. And now here's a pedigree or ancestry tree showing the inheritance of a Y-linked trait. So the legend here, so the circle is the female, and then the square is the male, but then the blacked out square is male with Y-linked trait. So what's going to happen is every son of this is going to be having this trait. So then there's going to be straight across there, and it's again all these straight across over there. But then on the all the other kids aren't going to have it. So the daughter's not going to have it, and then all this other one's not going to have it. And this one here, none of these are going to have it unless it's a Y-length over there that's that reproduced with a Y-length. And then on this side, yeah, none of these are going to have it. It's going to keep going straight down. And likewise, this one keeps going straight down. So that's fascinating stuff here. Let's erase this again. All right, so now let's continue further. Now let's look at a Punnett square. Or a Punnett square, and this can be used to predict the results of a test cross. We'll get to those in a bit. The chromosome theory of inheritance, which states that genes are found on chromosomes, was supported by Thomas Morgan's experiments with fruit flies, which established the sex linkage between eye color and sex in these insects. So in humans and other mammals, e.g. dogs or i.e. dogs, it is not feasible or practical to conduct test cross experiments, at least publicly. Yeah, but it might take a long time and a lot of ethical considerations, but nonetheless, privately or classified experiments, you better believe that's happening. Instead, pedigrees, which are genetic representation of family trees, are used instead to trace the inheritance of a specific trait or disease through multiple generations. Alright, so now here's an example. Punnett square depicting a cross between two pea plants, heterozygous for purple, capital B, and white, lowercase b, blossoms, or just flowers, etc. So it's pollen, the male, pistil, that contains the female organs, or the gametes, etc. And we'll get to those later when we discuss plants. But just an inheritance, so let's say it has it's heterozygous for purple, and then the white is recessive. And the purple is dominant. So it has a B and a lowercase b. And this one has a B and a lowercase b as well. So what you're going to have is you're going to intermix and you get 4. So this is BB, and then B, capital B, lowercase b, and then this one, capital B on this side, lowercase b there. And then likewise here, you're going to have a lowercase b and b. So then when you have 2 recessive, you get a white. Everything else, you get a purple. Like that. That's fascinating stuff. Yeah, so you could use these to predict cross family trees and so on. Then once you determine one is dominant, then you could predict what their offspring would be. MES note, in a test cross, the individual in question is bred with another individual that is homozygous for the recessive trait, and the offspring of the test cross are examined. So you can do a test cross where this one is homozygous. So you could test cross with this right here. Yeah, so you would first have to find out which one, if it's in fact homozygous right here. Yeah, for the recessive, and then you could utilize that to learn a lot about the different types of genes that a partner would have. So another plant, and then compare it with this, and so on. It's just fascinating stuff. So use that as a test cross, as you know this one would be a homozygous, and go from there. So the Punnett square is a square diagram that is used to predict the genotypes of a particular cross or breeding experiment. It is named after Reginald C. Punnett, who devised the approach in 1905. Let's continue further here. This is another test cross here. This is another Punnett square right here. Make it smaller. And here I just made it really small right here because it's pretty easy to see. So Punnett squares, a square diagram for predicting genotypes. Yeah, I already had this there, but keep it anyways. Showing typical test crosses and the two potential outcomes. Yeah, so these are test cross. Notice how these are the y, lowercase yy. Those are 2 recessive. So you have 2 lowercase recessive, and then you could analyze what the genes would be on this particular yellow corn and this other yellow corn right here. So the individuals in question may either be heterozygous, in which half the offspring would be heterozygous and half would be homozygous, recessive, or homozygous dominant, in which all the offspring would be heterozygous. Yeah, so in other words, this one here, if this is heterozygous, then what you're going to have is a homozygous dominant. Now, actually, no, that's this one here. So if you have it over here, if this is a dominant dominant on both sides. Here you're going to have a heterozygous where all of these will be heterozygous. So there isn't a YY in any of these ones here. It's going to be a uppercase Y, lowercase y. So just compare this test cross right here. So lowercase y and y for the green, and then you'll have this one here has a dominant yellow, but it has a recessive green there for the y. So you're going to have you're going to have it over here, which is a heterozygous 2 of these, and then the other half will be recessive dominant. No, I mean homozygous dominant. So you have this is a heterozygous right here, which is always going to be the dominant, and then you have a homozygous recessive. It's homozygous recessive because they're both the green alleles. Yeah, and you can see this right here. So y goes all the way across here, and this y across there. And then if you have this setup right here, where you have y on this test cross, and this has a y and y there, and you can just analyze it here. So if every single one of these would be green, then you know you're dealing with a capital Y here. So a dominant, homozygous dominant. Yeah, you can see that. So homozygous dominant, bold that, would have all the offsprings would be heterozygous. So you can learn that just by using this test cross, so this green one. And then this test cross, you can see that if there is some other green ones, you're going to know that this was actually a heterozygous, heterozygous. And heterozygous has a dominant and recessive. It's fascinating stuff.